In the spring of 2014, a wildlife research team working in the Allegheny National Forest in northwestern Pennsylvania collected 17 hair samples from a series of tree rubs along a remote ridge trail. The collection was routine. The team was conducting a black bear population density study for a conservation nonprofit based in Pittsburgh, and tree rubs, places where animals press and drag their bodies against bark to mark territory and relieve irritation, are standard collection sites for non-invasive genetic sampling. They bagged the samples, labeled them by GPS coordinate, packed them in a cooler, and drove them to a mid-sized university forensic biology laboratory in western Pennsylvania that had a standing contract with the nonprofit for wildlife DNA analysis. 6 of the 17 samples came back as black bear within 3 weeks. 4 came back as white-tailed deer. 2 came back as raccoon. 1 came back as domestic dog, which surprised no one given the proximity of private land to the collection site. The remaining 4 samples did not come back at all. 3 months passed. The nonprofit's field director, a woman named Carol Ashby, who had been coordinating wildlife research in the Allegheny region for 11 years, sent a follow-up inquiry to the lab. She received a reply saying the samples were still being processed and that she would be contacted when results were available. She noted that this was unusual. Standard turnaround for the lab's wildlife panel was 3 to 4 weeks. She filed the reply and moved on to other work. Another 3 months passed, 6 months total since submission. Carol Ashby called the lab directly. She was put through to a graduate researcher she had not spoken with before, a young man who stumbled through the conversation in a way she described to us as someone who had been told what not to say without being told what to say instead. He told her the samples had presented some complexity in the sequencing process and that the lab director would be in touch shortly. He ended the call before she could ask a follow-up question. The lab director called her the following morning. He told her that 3 of the 4 outstanding samples had been determined to be degraded beyond usable analysis and that results would not be available for them. He told her the 4th sample had returned a result consistent with a large North American mammal, but that the specific species classification was inconclusive due to database limitations. He told her the lab was closing the file. He thanked her for her patience. Carol Ashby asked him what large North American mammal the sample was consistent with. He said he was not in a position to elaborate beyond the summary he had provided. She asked him if she could have the raw sequencing data for the 4th sample so she could have it reviewed independently. He told her the raw data was proprietary to the lab's internal process and not available for external distribution. She asked him to send her the physical samples back. He told her they had been disposed of as per standard laboratory protocol for samples past their retention window. 6 months and 4 days after submission. Gone. Carol Ashby has a background in wildlife biology and enough familiarity with genetic analysis to understand what she was being told and what she was not being told. She sat with the phone call for 2 days before deciding what to do about it. Then she drove back out to the ridge in the Allegheny National Forest, where the original samples had been collected, and spent 3 days looking at the tree rubs with fresh eyes. What she found on that return visit, in the soft soil at the base of a large white oak at the end of the rub line, was a footprint. She almost missed it. It was partially obscured by leaf litter and had been degraded by 2 weeks of early spring rain. But the outline was clear enough. Bipedal, wide across the ball, deep heel impression. She measured it with a tape measure she kept in her field kit, 14.5 inches in length. She looked for more prints on either side. She found 2 additional partial impressions, suggesting a stride interval of close to 4 feet. She had been working in Allegheny bear country for 11 years, and she knew bear track morphology the way a mechanic knows engine sounds. What she was looking at was not a bear track. She took photographs with her phone and a small field camera. She did not cast the print. She has said since then that she wishes she had, but in the moment, She was still processing what she was seeing, and the instinct to document photographically overrode the instinct to preserve. She walked the rest of the rub line and found 2 more partial bipedal impressions in a muddy drainage crossing approximately 80 meters to the northeast. Same morphology, same size. She drove home and uploaded her photographs to her laptop. She sat at her kitchen table in Clarion, Pennsylvania, and looked at them for a long time. Then she opened her email and started writing to every contact she had in the wildlife research community who she thought might respond without dismissing her. Most did not respond at all. 2 responded with politely worded suggestions that she consider alternative explanations. 1 responded with genuine interest. That one response changed the direction of everything that followed. The person who responded was a molecular biologist named Dr. Thomas Varon, then a postdoctoral researcher at a genetics institute in the Mid-Atlantic region, who asked that his institutional affiliation not be identified in this documentary for reasons that will become clear. Dr. Varon had been quietly building a personal database of anomalous wildlife DNA samples for 4 years. Not as an official research project, as a private interest that he pursued on his own time using his own resources, aware that it would not survive formal academic scrutiny and not willing to risk his career by making it one. He had 7 samples in his database from 6 different states, all collected by credible field researchers, all returned from standard wildlife labs as either inconclusive or unclassifiable. He had been contacted about 2 of them by the submitting researchers, who, like Carol Ashby, had found the non-answers they received more troubling than a clear negative would have been. When Carol Ashby described the 6-month delay and the lab's refusal to return raw data or physical samples, Dr. Varon told her it was the 3rd time he had heard a version of that specific sequence of events. A delay of between 4 and 7 months, an inconclusive result offered without supporting data, a refusal to return samples citing disposal. The pattern was not random. Patterns are never random when they repeat across unconnected institutions and unconnected researchers. He asked her if she had any additional material from the original collection site. She told him she had not kept any, that all samples had been submitted to the lab. But she told him she was going back out to the ridge, and she was going to collect everything she could find. She went back in late April of 2014, 2 weeks after the phone call with the lab director. She spent 2 full days at the site. She collected 6 new hair samples from the same rub tree and 2 adjacent trees, using sterile collection tubes and a protocol Dr. Varon had emailed her the night before. She documented every collection point with GPS and photography. She found 3 more partial footprint impressions in a second drainage crossing, which she measured, photographed, and this time cast using plaster of Paris she had driven 40 minutes to buy from a hardware store in Warren, Pennsylvania. The casts are in Carol Ashby's possession. She showed them to us during filming at her home in Clarion. They are stored in a plastic storage container under her desk, wrapped in cloth to protect the detail. She lifted them out carefully and set them on the kitchen table. The largest cast measures 14 3/4 inches in length. The width across the metatarsal region is 6.5 inches. The heel is defined and rounded, not elongated, as it would be in a plantigrade bear print. There are 5 distinct toe impressions across the front of the cast, with no claw marks. The depth of the impression in the original soil, which she documented photographically before casting, was consistent across the full length of the print, indicating a weight distribution pattern that matched bipedal locomotion rather than quadrupedal movement, transferring onto a hind foot. Dr. Varon reviewed high-resolution photographs of the casts remotely. He is not a physical anthropologist, and he was careful about the limits of his assessment. But he noted that the morphology was consistent with descriptions in the existing literature on anomalous large bipedal track casts from the Pacific Northwest and Gulf Coast regions. He noted specifically that the mid-foot pressure ridge, A raised band of compressed soil running laterally across the middle section of the cast was a feature documented in several significant cast collections and considered by some researchers to indicate a flexible mid-tarsal joint not present in the modern human foot. He said he could not draw conclusions from photographs alone. He said the casts needed to be examined in person by someone with relevant expertise. She sent the 6 new hair samples to Dr. Varon by overnight courier. He processed them himself in a university laboratory over a weekend, using equipment he had legitimate access to as a postdoctoral researcher, running the samples against both a standard wildlife species database and a secondary database he had compiled from published primate genomic sequences. He ran the analysis twice to verify the output. 4 of the 6 samples were standard: 2 black bear, 1 white-tailed deer, 1 unknown conid consistent with coyote. The remaining 2 samples returned a result that he sat with in that laboratory on a Saturday afternoon in May for a long time before he wrote it down. The mitochondrial DNA sequence from both samples showed a primate haplogroup. Not bear, not ungulate, not conid. Primate. The sequence was consistent with the superfamily Hominoidea, the group that includes gibbons, great apes, and human beings. But when he ran the sequence against every published hominoid reference genome in the database, the match confidence did not exceed 89% for any known species. For context, a human hair sample run against the human reference genome returns a match confidence above 99.9%. An 89% match to the nearest known relative means the sample belongs to something that diverged from that relative long enough ago to accumulate significant genomic distance. He ran the analysis a 3rd time. Same result. He ran it on each sample independently. Same result from both. He closed the laboratory that evening, went home, and did not tell anyone for 11 days. He has explained that 11 days as the time it took him to decide whether he was willing to accept what the data was telling him. He is a scientist. His entire professional identity is built on following evidence to conclusions, not deciding in advance what the conclusions should be. He had followed the evidence. The evidence had arrived somewhere he had not planned to go. He spent 11 days making sure he was not making a mistake. He wasn't making a mistake. On the 12th day, he called Carol Ashby and told her what he had found. She was quiet on the phone for a long time. Then she asked him what he needed next, not what it meant, not whether he was sure, what he needed next. He has said since then that her response in that moment is part of why he trusted her with the rest of it. What he needed next was a second independent laboratory confirmation from a facility with no connection to the first lab and no institutional stake in the result. And that, it turned out, was considerably harder to arrange than either of them had anticipated. Here is part 2 of your script. Approximately 10,000 characters of pure vidrush-ready prose. Dr. Thomas Varon spent 3 weeks contacting independent laboratories capable of running advanced mitochondrial DNA sequencing. He was not looking for a lab that would confirm his result. He was looking for a lab that would run the analysis blind, without knowing what the previous result had been, and return whatever the data actually showed. He contacted 7 facilities. 4 declined without explanation. 1 cited capacity constraints. 1 expressed interest, and then stopped responding to emails after he sent the sample details. The 7th, a private genomics company in the Research Triangle area of North Carolina that did contract work for pharmaceutical and agricultural clients, agreed to run the analysis for a standard commercial fee with no questions about the source material. Dr. Varon submitted the 2 hair samples under a project name that gave no indication of their origin or the nature of the previous analysis. He paid the fee from his own account. He listed himself as the contact under a name variation that would not connect to his published research profile. He waited. The Research Triangle Lab returned results in 18 days. Dr. Varon opened the result file on his laptop at 7 in the morning before he left for the university. He read through the sequencing output twice. Then he closed the laptop, sat at his kitchen table, and called Carol Ashby. The second lab's analysis was consistent with his own. Primate haplogroup, Hominoidea superfamily. Match confidence to the nearest known species below 90%. The two labs had used different sequencing platforms and different reference databases. They had arrived at the same place independently. The result was not a sequencing artifact. It was not contamination. It was not operator error. It was real. It was consistent. And it meant that 2 hair samples collected from tree rubs on a ridge in the Allegheny National Forest in Pennsylvania belonged to a primate that was not in any published genomic database on Earth. What Dr. Varon and Carol Ashby did next is the part of this story that most clearly illustrates the gap between what science is supposed to do and what science is actually able to do when the data points somewhere institutionally inconvenient. They tried to publish, not a sensational claim, not a press release, a properly formatted, methodologically rigorous scientific paper documenting the collection protocol, chain of custody, sequencing methodology, and results, submitted for peer review to a mid-tier journal covering wildlife genetics and molecular ecology. The first journal rejected the submission without sending it to peer review. The editor's note said the subject matter fell outside the journal's scope. Dr. Varon read that note several times. The journal's stated scope included, in its own language, the genetic characterization of novel and understudied wildlife populations in North American ecosystems. He wrote back, asking for clarification on why an unidentified North American primate sample fell outside that scope. He received no response. The second journal sent it to peer review. 2 reviewers returned comments. The first reviewer's comments were substantive and engaged with the methodology directly, raising questions about contamination controls and reference database completeness that Dr. Varon considered fair and addressable. The second reviewer's comments were 3 sentences long. They said the premise of the submission was not consistent with the current scientific consensus on North American megafauna, and that publication would not serve the journal's reputation. The editor rejected the paper on the basis of the second reviewer's comments, without addressing the first reviewer's substantive engagement. The third journal never responded at all. Dr. Varon posted a preprint of the paper to a public academic repository in September of 2015, 16 months after Carol Ashby first collected the samples. Within 48 hours, the preprint had been downloaded 4,000 times. Within a week, it had been cited in online discussions across wildlife research forums, cryptozoological communities, and several mainstream science communication platforms. Within 10 days, Dr. Varon received an informal communication from a senior faculty member at his institution, suggesting that his association with the preprint was creating conversations at the administrative level that he might want to be aware of. The faculty member was not threatening him. The faculty member was, in the way of academic institutions, informing him of which way the wind was blowing. He took the preprint down 2 weeks after posting it. He has not attempted to republish the paper since. He still has the original data. He still has his own sequencing results. He still has the Research Triangle Lab report. They are stored on an encrypted drive that he keeps in a fireproof lockbox at his home. He showed us the lockbox during filming. He did not open it on camera. He said he was not ready to make the contents public in their entirety, but that he wanted their existence documented. He also said something during that conversation that we have thought about since. He said that the thing that troubles him most, more than the journal rejections, and more than the institutional pressure, is the original laboratory's behavior. The one that held Carol Ashby's first samples for 6 months and then disposed of them. Because Dr. Varon, in his 3 years of collecting anomalous sample reports before he connected with Carol Ashby, had documented 2 other cases where a standard wildlife lab had held samples significantly beyond normal turnaround, returned an inconclusive result without supporting data, and then reported disposal before the submitting researcher could request return. 2 other cases, 3 if you count Ashby's, all from different labs, all in different states, all involving samples collected within 200 miles of documented high-density encounter corridors. He does not believe in coincidence as an explanatory framework when a pattern repeats with that kind of structural consistency. He said that clearly. He is a scientist, and he said it clearly. The Allegheny National Forest sits in the northwestern corner of Pennsylvania, covering approximately 513,000 acres of mixed hardwood and conifer forest, in a region that has produced some of the most consistent, long-term cryptid encounter documentation in the eastern United States. The BFRO database lists 74 reports from Pennsylvania as a whole, with a notable concentration in the Allegheny Plateau region covering Forest, Elk, and McKean counties, the precise counties that contain the Allegheny National Forest. That cluster of reports spans 6 decades of documentation and includes visual encounters, vocalization events, track finds, and 3 separate instances of tree structure formations, deliberately arranged branch and sapling configurations that field researchers associate with territory marking behavior in several encounter areas across the country. One of those tree structure reports, filed in 2011, 3 years before Carol Ashby's sample collection, came from a wildlife photographer named Marcus Denner, who was documenting old-growth tree canopy conditions in the northern section of the forest. Denner found a series of bent saplings arranged in an X formation at a trail junction near Tionesta Creek, photographed them, and submitted them to the BFRO with coordinates. Those coordinates place the formation approximately 2 miles from the ridge where Ashby's tree rubs were located. Denner is not connected to Carol Ashby. He did not know about her work when he filed his report. He filed it because he could not explain what he had seen, and he thought someone should know. The global context for anomalous primate DNA evidence is limited, but not absent. In 2012, a large-scale genetic study led by Brian Sykes, a professor of human genetics at the University of Oxford, collected hair samples attributed to Bigfoot, Yeti, Almasty, and other cryptid primates from repositories around the world, and subjected them to rigorous mitochondrial DNA analysis. The study, published in the Proceedings of the Royal Society B in 2014, analyzed 36 samples. 34 were matched to known species. 2 were not matched with confidence. Sykes described those 2 samples as presenting unusual haplotypes that warranted further investigation. He specifically noted that one sample, collected in the Himalayas, showed a sequence consistent with an ancient polar bear lineage thought to be extinct, raising the possibility that relic populations of archaic animals persist in remote ecosystems beyond the reach of current biological surveys. The study was widely covered as having debunked cryptid hair evidence. That coverage consistently omitted the 2 unresolved samples and Sykes' own statement about warranted further investigation. Sykes himself said in a subsequent interview that his findings had been misrepresented and that his actual conclusion was that the field warranted serious scientific attention, not dismissal. He died in 2020. The 2 unresolved samples from his study remain unresolved. In 2013, a separate DNA study led by Melba Ketchum, a veterinary geneticist in Texas, claimed to have sequenced nuclear DNA from purported Bigfoot samples and found evidence of a novel hominin with a human maternal lineage and an unknown paternal lineage, suggesting a hybrid species that diverged from modern humans within the last 15,000 years. The study was published in a journal of limited circulation and was immediately and widely criticized for methodological problems, including contamination concerns and insufficient peer review. The scientific community's rejection of the Ketchum study was rapid and largely justified on technical grounds. But several researchers noted that the underlying premise that an anomalous primate population might carry a genomic signature showing partial human ancestry was not inherently implausible, given what is now understood about archaic human interbreeding with Neanderthals and Denisovans in the Pleistocene. The science of ancient DNA has shown repeatedly in the last decade that the boundaries between human and near-human species in the Pleistocene were more permeable than anyone previously assumed. Dr. Varon is aware of both the Sykes study and the Ketchum study. He does not align himself with either. He says his data is his data. He is not building a theory. He is documenting a result. The result is what it is, regardless of the theoretical framework applied to it. Carol Ashby still works in wildlife research in the Allegheny region. She has not returned to the specific ridge where she collected the original samples, though she has worked in adjacent areas. She keeps the plaster casts under her desk. She keeps a folder on her laptop of every photograph from the 2 collection visits, backed up to an external drive and a cloud account. She described her state of mind about the whole episode as a kind of permanent open file, something that cannot be closed because it was never resolved, and the evidence that would resolve it is gone, disposed of by a laboratory that gave her no satisfactory account of why. She said that the thing she thinks about most often is not the footprints, and not the casts, and not even the DNA results. She thinks most often about the original lab's behavior, about the 6 months of silence, and then the call from the lab director who told her just enough to end the conversation, and not enough to answer any question she actually had. She said that whoever made the decision to bury those results, to hold them for 6 months, and then dispose of the samples before she could retrieve them, that person knew exactly what they had. You do not dispose of inconclusive samples. You return them, or you hold them for further analysis. Disposal means you made a decision about them. Disposal means something was found. She said that quietly, without drama, the way someone says a thing they have been certain of for a long time. The Allegheny National Forest is open to the public. The ridge where the tree rubs are located is accessible on a marked trail. The white oak at the end of the rub line is still there. Carol Ashby checked last autumn. The rubs are still being used. She does not know by what. She did not collect samples. She said she did not see the point of collecting samples. She could not trust a laboratory to process honestly. She looked out the window of her kitchen in Clarion when she said that. The Allegheny Plateau was visible in the distance, dark and dense in the gray November light. She looked at it for a moment, and then looked back at the camera. She said, "It knows we found it. Whatever it is, it knows. Because whatever it is, it has been watching this forest a lot longer than we have been trying to figure out what it is." She picked up her coffee cup. She did not say anything else. Outside the window, the forest held the ridge line the way it always has. Still, patient, indifferent to what we can prove and what we cannot. Keeping, as it always has, its own account.